Hey guys, welcome to our channel. So, we've been decoding the agreed upon section of Bible changes, and we've got through the letters in all the categories. That would be finance or stock market, genetic, medical, chemical, um, research groups, technology, and random. And in random, basically, by the time we hit those, we're looking for different stuff. We're looking to see if anything else, any other category surfaces and comes up over and over that we feel we need to run as well. And we've gotten through the letters. We've converted them. We've converted those letter extractions to numbers using a numerology chart. And we've got the numbers to run forwards and backwards in finance in blocks of 4. Um, which was what I decided I was going to try. And if it didn't go through, then I was going to note that and then see if I could make it go through. But I was trying to make it not go through in a certain way, and blocks of 4 look about right here to either go through or not. And important to note once again that in some of the shorter decodes, groups of 4 did not go through. I had to back it down to 3. In some cases, even 2. But in this case, we were trying to see if we could get it to not go through. It's a longer decode. In a shorter decode, 3 might be your magic number, pre-selected. I'm not sure, guys. And also in trying to scrutinize harder, I find myself struggling. I don't want to leave information on the table. And I found that I am leaving information on the table by mistake because I kind of got off on my search. And then I was searching a different sequence of 4 because I had skipped a letter or a number, and it was going through just fine. And by the time I realized I had skipped a sequence and went back to make it go in blocks of 4 or not, it went right through anyway. So, and so we're leaving information on the table just doing blocks of 4. All right. But we're also we're scrutinizing harder, and that's another thing to mention. Also, in doing that, I found myself wanting to throw out information, and I realize that I can't. If the information's there and the information seems relevant, we got to include it. Whether I want to, now I've got myself wanting to throw information out just because, but I can't necessarily do that, or else I'm skewing the results the other way. So we're trying to. So we're gonna once again, you guys can determine what you find relevant if we provide these searches and what's not relevant. But if you start off with 955, oh, are your numbers, guys? These are your numbers. I'm not even gonna read them off. I will just show you the numbers in this case. Try to get them all in there. All right, and you can see they start here and go all the way to the end, and then they come back and start here and go all the way to the end in order. And if you wanted to see them correspond, I'll provide the link which we got these guys and explain how we did it. But if you wanted to see them corresponded to their letters, it starts here and then comes back. All right, so you got all that. All right, so now we're into genetics. All right, and if you start with 9556, you get ATP5MPL gene ID 9556, and it's a protein coding gene. But that's not all you get. You get You get a couple of gene IDs here. You get a variation ID, which is a M5521G greater than a simple variation report. Your variation ID is 9556. You also have your number right here in vector medical reverse genetics system. Allow the generation of influenza A virus entirely from cloned cDNA. However, since, alright, so you're dealing with virus cloning there. Well, I don't know why they'd ever want to do that. You also get a gene 9556, ATP5MPL gene, and it's gene interest gene 9556. And so some of these the number isn't there. Some of these the number's right there. Your number code. Study of genetic diversity in salt and safflower genotypes. All right. So you got several things: natural selection and genetics of adaptation in 3-spine sickleback. All right. So you got several things going on that could be relevant. I'll provide the search, guys. But if you move on from that, you get 1765, and that ends up being Encyclopedia of Forest Sciences. Remember, in National Wildlife Federation came up over and over and over. Well, Encyclopedia of Forest Sciences is right here. It's in a book. The numbers there. I don't know how significant it is, but beyond that, I found that significant because of the NWF coming up over and over and over through all the categories. The National Wildlife Fund. But you got a few other things. You got pages in a Human Molecular Genetics, Volume 2, Issue 11, 1 November 1993, pages 1765 through 1772. And I wanted to throw that out. I really did. Wanted to say, no, that's just some pages in a book. But who knows what's within those pages? So you almost have to include that as something that could be calculated. And it goes on, guys. There's more. You got more seeding stuff, more seed stuff. You got extending the rapeseed gene pool, rapeseed, RAPESEED, rapeseed gene pool with. That sounds terrible, but that's what it says. Oilseed rape breeding material. All right. I don't think I like that term, but that's what it's termed, and that's what they got. So, nothing I can do about that. I'm sure it has an entirely different meaning than your traditional word for that. I'm sure it has everything to do with the genetics therein. But you got several things on that, and if you move on from 1765, you wind up with 5391, which ends up being NIM_030621 dot dicer or dot 4 dicer 1 C dot 5391G. 5391. And this is an allele. And it has a different ID, but the number's right there in its name, which is a really weird long name. And there's several of these, guys. There are several of these that come up. But if you get past that, you have gene regulation work earns chemistry prize. Science magazine. I don't know that that's even relevant. Abstract 5391. That's what I was looking for. Low frequency variant detection. Molecular and cellular biology genetics. Abstract 5391. Low frequency variant detection and tissue of origin. Exploration using liquid biopsies, liquid biopsies. So that could very easily be irrelevant and calculated. There's your number. Now how about an introduction to genetic epidemiology? Fearful chickens and worried mice, shared genetic influences. And there's the number. So that's just page 1, guys. You got to watch for the number not to be there on occasion, and some of the stuff I would now throw out, especially knowing what I know now. But you never know what's relevant and what's not relevant. So, again, it's still just as big of a guessing game as it always has been. I'm just scrutinizing a little heavier is all. So if you move on from that, you actually next have 7111, and if you end up with NM underscore 3 zeros 426.3 LAMA2C.7111T. All right, and that is that's genetic diagnostics. So you have MIR 7111 gene as well. If I'm not mistaken, that is a non-protein coding RNA. You also have medical genetics test details, Baylor Genetics Laboratories, test code 7111. So you got a test code, and that seems interesting because that comes up some more as we go through here. You also have, do I see my campus box 7111? So yes, it does bring you to science in the movies, genetic engineering, and gene editing. I don't know what the movies have to do with gene editing and genetic engineering, but whatever, it's there and it may or may not be important. Pediatric and Medical Genetics Services PC 7111. So you got a few things going on with even 7111. MicroRNA 7111. So how about microRNA? You also get TMOD1. There's your number, 7111. So you get quite a few things out of that. And if you move on from that, you get you get to 5142, and 5142 ends up dealing with lung small cell cancer, Atlas of Genetics and Cytogenics, Atlas ID 5142. So you're dealing with cancer there in genetics, Atlas of Genetics. Definitely could have been calculated. Here's some more Atlas of Genetics, 5142. You also have NM underscore 000138.4 FBN1 C5142G. All right. So yeah, you got a few things going on with that. Plasmid encoding, a combination of mosquito larvicidal genes. There's 5142. P4BDA 5142. Yale School of Medicine as well. You got faculty genetics, Yale School of Medicine. So you got another test code 5142. And so, that's that's one page, and it goes on. I'll provide the link, the search, guys. Moving on from that, you have 8582, which ends up being, you got a few interesting things here, actually, guys. First of all, you got NM underscore 004006.2 DMD C.8582T. And that's an allele description, variant report. So it's a variant. You also get, I don't know that I see my number there. Dismissing it, no number. Dismissing that, no number. Don't really think I can dismiss editorial board DNA research, Oxford Academy, Oxford Journals, and there's your number. Genetics of movement disorders, and there's your number. Got bacteria collection, NCTC 8582, so there's your number. The theory of facilitated variation, there's your number down there. Can't really throw any of that out, guys. And there's Institute of General Genetics and Cytology, and there is 8582 right there in the web address. So, and that's just one page of that stuff. So I'll provide the search, guys. Could all be relevant. So if you move on from that, you wind up with 5171. Maybe I didn't write it down. We got 5171. So I just didn't write this down, guys. But yeah, okay. So next you have 5171, which ends up being one of those genetic variants, C5171T. There's one of your variants. You also have UAB, Center for Community Outreach Development Genetics, 5171. That's a phone number. Can't really throw that out. Molecular phylogenetics of genus Trichosporin infrared. And there's your number, 5171 down there, so I can't throw that out. You also have ORCID, genetic diseases following benchtop exome sequencing using the Ion Proton Journal of Genetics. Can't really throw that out. Got more ORCID stuff. 5171. So yeah, there's my number again. Gallus. Something I can't pronounce. UK Genetic Testing Network, guys. Introduction to Veterinary Genetics. Okay, so there's some more of your animal genetics that my fiance predicted in the beginning of all this. Introduction to Veterinary Genetics. And that it was predicted. So, yeah, guys, a lot of things going on with this. And if you move on from that, you end up with 1117. And 1117 ends up being long LINC 01117, long intergenic non-protein coding RNA, RNA 1117, Atlas of Genetics. You also get suppression of mitochondrial tRNA gene mutation phenotype. And there is her number, 1117. You also get strain VC2597, genotype Y113G7B.17K or GK1117. So there's your 1117. That's a strain of something. probably a virus. You got quantitative classification and natural clustering. That's more genetic stuff, and there's your number. Drosophila rhino encodes a female-specific chromodomain protein. And there's the number. You got some autism stuff here, sex-specific risks. And you got 1117 there. You got population genetics of endangered Mohegan skate. Flybase reference report here as well. So you got quite a few things going on with 1117. And if you move on from that, you have 1951. Well, there wasn't a whole lot there. In fact, I thought we were going to say it didn't go through. But I can't throw out statistical methods in genetics 1951. Statistical methods in genetics, it's on PubMed. So there again, we found something that seemed relevant, like it could be calculated. And so we had to pass it. And I can't even say that barely scraped by. That's fairly significant, statistical methods in genetics. So, yeah, it goes right through. Then if you move on from that, you have 2142. You have genotype GLP-1 E2142 plus just molecular genetics of a biological clock in Drosophila. You got strain JK298 genotype, GLP-1 E2142. I think I read that one already. You have another strain genotype with OK2142. And you got all sorts of strain genotypes. Well, let me look at that again. 2142. And the other ones, I don't see my number, guys, so just the 2142. Then it goes on to several others. Strain. 2142. I do too see it. RB2142. OK2142. VC2142. So yeah, they are all there, guys. Strains and genotypes. You got African elephant conservation and population genetics 2142. Oh, more animal stuff. Just like my fiance predicted. All right, and you've got a few of those in here so far going on. So I guess it was predicted. So it's hard to throw any of this out when my fiance predicted this, and there it all is. There's the animal stuff she predicted. So if you move on from 2142, you get 9412. And that is MED21 gene ID. Let's see, MED21. Yep. You come up here, you got mediator complex subunit 21. That's what it is. MED21, mediator complex subunit 21, Homo sapiens, human, gene ID 9412. You got advances in rice genetics. I don't know how relevant that may be, but it's in there, and the number's there. It's hard to throw it out. I can throw that out because I don't see my number. But here's some microRNA 172. And you have 9412 right there, down below. So you got a couple things going on with 9412. If you move on from that, you got 3633. And 3633 winds up being 3633 winds up being one of those gene variants, C3633DUPT. Duped. But no, really, that's what you got. It's one of the gene variants. I'm trying to see if my number's here. 3633. All right, critical folding disease and medicine genetics and genomics news. All right. You also have variation OK3633, species CAENORHABDITIS, elegans. That's a gene variation. Synergistic effects of genetic variation in nicotinic, nicotinic, nicotinic. There's the number right there. You also have volume 143, issue 3633 of Nature. And you have the National Wildlife Fund, my fiance's prediction of the animal genetics. So you got a few things going on with 3633 as well. And if you move on from that, you have 8595. And 8595, you wind up with several things going on. There's your number. So MIP or MIIP, migration and invasion inhibitory protein, HS958 or 8595. Targeting an invasive pest with gene drive. Do I see? Yeah, 8595. Right there's my number. There's animal stuff, so pest nonetheless. But you got genetic articles, bacteria collection, NCTC 8595. So you got a few things going on with 8595 also. And then you get to, you move on from that, you get 2951, which ends up being a worm-based gene. 21UR2951. Oh, more animal genetics. It's worm base. You also have medical genetics test details, Baylor Genetics Laboratories, test code 2951. So there's another one of the test codes from Baylor Genetics Laboratories. You got a couple of those actually. You might have had 3 altogether, but definitely 2. And then you're left with your last 5 letters, which I broke into blocks of 4, because that's what we said we were doing, blocks of 4. And you have 2622, and 2622 ends up coming up with locus ID NCBI 2622. You've also got one of those genetic variations, pt.1 2622. Couple of times. You got strain RB2622, genotype GCY-27. So you got another one of those strains. Department of Behavioral ecology and evolutionary genetics. There's 2622 right there. And we've had behavioral stuff in this particular decode in the letters, if you recall. Some behavioral genetic studies and some behavioral national behavior-based natural interest waivers, right? And if you move on from that, then you're left with 6223, and that ends up being 21UR6223 gene, and it's worm base. So more of your animal genetics. You also have 6223 Atlas ID 177. It is RPS19, ribosomal protein S19. So that's some genetic stuff. You got another one of those variants, 62C.6223G. I'll just provide these searches, guys, because there really is a lot of stuff, and some of it's going to be relevant, some of it isn't. And there's so much of it that I guess, if you guys want to, you can determine for yourself what's relevant. You've also got cancer risk, role of environment. So here's some more cancer stuff. Issue 6223 in Science Magazine. So that actually, and it goes on, guys. But that gets you through going forwards in genetics. And holy cow, did we run long? But that's what's there. And so that's all there. So we'll run backwards next. Hopefully that can go faster. In genetics, and then it'll be on to medical. See what's there and what isn't there for that. But so far, this is going through just fine in our blocks of 4. But will that be the case in every category or even backwards through this one? I don't know. Maybe not. If not, we're going to make a note of it, that it didn't do it. And we'll see how it does go through. So that's something we're going to be looking at in the future. So just more information, more data, which is what we're collecting. Hard data on the Mandela Effect. And hopefully we can do something good with this data. It would be our hope to be able to do something good, figure something out. And, we've only scratched the surface, so who knows? Who knows? And there's a lot left to learn. So that's forwards. We'll come back next in genetics. And so for now, guys, thanks for all your thumbs up, thumbs down. Thanks for your comments, leads, feedback, and subscriptions. And thanks a lot for watching, guys. And for now, you guys have a great rest of your day, and have a happy and safe new year if I don't come out with another video until then. All right, have a good night, guys.